Can somebody state whether you hear me or not?
Yeah, I can hear you.
Great. Let me share my screen. All right, you should be seeing my screen now. Let's go to the syllabus. So today we're going to talk about Lab 00, lab safety training. In the real lab, why did that happen? Oh, maybe that's the lecture website finished. Let me take a look here. Yeah, it's the lecture website. All right, great. My computer will speed up because Zoom isn't making the lecture file now. Um, In the real lab, you would actually go into the lab and I will discuss lab safety training. For here, we will just go over lab safety training for your quizzes. Uh, Following the same syllabus, so I'm not going to go over most of this material. Let me do state that we do have one optional textbook, which is the Photographic Atlas for the Microbiology Laboratory by Leboff and Pierce. It does provide photographs of microorganisms and all the tests we're going to talk about in this class. So in that case, it would be good, but you may find pictures as well as information about them from other sites like online. So if you want to do a search, you can, or you can get the photographic atlas for the microbiology laboratory. Safety information for students and physicians. The following information is included only for educational purposes, as future nurses and dentists, dental hygienists will need to know these issues. You also need to know them for the quiz. Microbiology labs are operated at Biosafety Level 2 or BSL 2. That means protective equipment, lab coats, gloves, goggles when a splash hazard is used, are required to work in a real microbiology lab at Clark. Now, obviously, we're going to do everything online, so not everything will be biosafety level 2, but know that the lab at microbiology lab at Clark follows BSL 2 levels. Students who are immunocompromised, living with or caring for an immunocompromised individual and or pregnant or may become pregnant, caring for living, someone caring for infants should consult with their physician to determine the appropriate level of participation in the lab. And usually, somebody like that, we would specifically give them a microorganism to work with that would be considered, quote, safe, as opposed to something which would be at risk for somebody. Alternative combinations may be indicated. In the microbiology lab, only students in microbiology are allowed into the lab. You should monitor your symptoms, and if you come down with gastroenteritis or other symptoms caused by some of these organisms, then you should take that into your physician. We have biosafety level 1 organisms considered at lowest risk: Enterobacter genes, Enterobacter cloacae, Enterococcus durans, Escherichia coli, Pseudomonas fluorescens, Serratia marcescens, Micrococcus luteus, Micrococcus varians, Saccharomyces cerevisiae, Staphylococcus epidermidis, Streptococcus bovis, Lactococcus lactis, and Citrobacter florendi. We also have biosafety level 2 organisms which are considered medium risk. This will be Enterococcus faecalis, Rhodius mirabilis, Pseudomonas aeruginosa. It looks like we're missing one of them. Shigella dysentery, Shigella flexneri, Staphylococcus aureus, Streptococcus agalactiae, Streptococcus mitis, Streptococcus pneumoniae, and Streptococcus pyogenes. You must follow lab safety rules whenever you are to go into a microbiology lab. At Clark, we have these lab safety rules. For you guys, you only need to know them for the quiz. Let me state that safety is something that most nursing programs want their students to know or learn. So safety is of primary importance in the lab. Never take shortcuts which risk the safety and accuracy of the environment, others, as well as yourself. Let me write this up. You're not allowed to take any personal items to your lab bench or inside the lab. They must remain in your bag in a space provided for the bags, which we call the cubbyhole or the cubicles. All required equipment, paper, pens, will be provided for you and must not be taken out of the lab. Lab safety rules described in detail in the first lab, so we're going through it now. Only enrolled microbiology students and staff are permitted in the lab unless authorized by the instructor. All personal items, including but not limited to book bags, books, purses, cell phones, coats, must be stored in the designated cabinet or cubbyhole as soon as you enter the lab. This region in the door of the lab is considered outside the lab. So when you first walk into the microbiology lab, the door and the cabinets where you stick your stuff are considered outside the lab. You then walk into the lab, going to the sink to wash your hands. Once you wash your hands, you dry your hands, you should then put on the lab coat, put on gloves, and if you're going to be working with a splash hazard, you should put on goggles. Note, the goggles are only worn in the microbiology lab when there's a splash hazard you'll be working with. So most of the time, goggles are not worn in the microbiology lab. And then when you leave the lab, you take off your gloves, take off your lab coat. If you have goggles, take off the goggles and go over to the sink, wash your hands, and then walk over to your cabinet. And now you walked outside of the lab, pick up your things, and then you can leave the door of the lab. Any question about any of that?
Nope.
All right.
I'm sorry. Are we going to have actual classes in person?
No, no. Like I said, you just need to know this for the quizzes.
Oh, I understand. Sorry.
All right. If anything prohibited is brought into the lab proper, they will be confiscated for safety reasons and only returned after it has been autoclaved or cleaned with bleach. So if you were to take your phone to your table, it would be confiscated and then autoclaved or stuck in a bucket of bleach for 15 minutes before returned to you, in which case it would no longer work. Any question about any of that?
I had a quick question. I see in 2B that it states that you'll send yourself digital photos of lab notes. If you're not bringing your cell phone in, how is it that people are taking digital pictures and sending their lab notes to themselves?
In the lab, if you were to go into the real microbiology lab at Clark, we have what's that Apple computer? It's like a Apple Chromebook, something like that. Um,
Okay,
and you can take pictures with that, like of your notes, and then email them to you. And then you can study your notes or take pictures of photographs and take them out of the lab. Meaning you can take anything digital out of the lab, but obviously the computer stays in the lab at all times.
gotcha. Okay, so I'm just curious for any sort of that information that's not included here. I see that it does mention that uh, you are able to send digital photos of your lab notes to yourself or whatnot. Do we need to know these like in between things as to what's in the lab, like the that you had mentioned, like the setup for how to go through the process of digital photos and sending it to yourself via email, or do we just need to know the stuff that's on this document?
You need to only know the stuff that's on this document. I believe I'm not asking any questions about how students take a digital photograph and then that's how you take it. Now, obviously, if I ask a question like, how can you take material to study out of the lab? I don't think I asked that, but if I do, you would say you photograph it and email it to yourself, and that's how you can study your lab notes.
Okay, cool.
Okay.
Thank you.
Uh, if you were to um, wanting to use a phone, we actually have a phone in the lab. It's the old, what do you call it, push button type, and you can use your gloves on it. And if you have bacteria on your gloves, you can get it on the phone. This phone is kept in the lab. You can actually call out of the lab. You can also receive a call into that phone, and you would not need to leave the lab to use your phone. However, if you wanted to use your phone, you'd have to take off your gloves, take off your lab coat, go over to the sink, wash your hands, go over to your backpack in your cubbyhole, and take out your phone, leave the lab, dial someone. When you're done with that call, you then have to bring the phone back into the lab, or back into the room, I should say, put it in your backpack, go back to the sink, and now you've entered the lab proper, and wash your hands, go put on your lab coat, and then put on your gloves. So that would be the proper procedure for uh, using your phone actually outside the lab. All right, where's my mouse? Don't see my mouse. There it is. You are allowed to take some personal items into the lab if you are wearing them, such as jewelry, your clothes, watches, glasses. You should not touch these items that you are wearing. Any question about any of that? All right. All cultures you're working with are considered to be potentially pathogenic. A pathogen is something which causes disease. Keep hands, pencils, and other materials out of your mouth, ears, nose, etc. No eating, drinking, smoking, chewing gum in the lab is allowed. You are required to wear gloves and they are provided for you in the lab. You should monitor yourself for possible symptoms of an infection meaning getting diarrhea, fever, abdominal cramps, call your healthcare provider if you come down with any of these symptoms. A document with information about the bacteria. It's a 4-page document. It's titled Information for Students and Physicians, provided on the class website. You must wear proper attire in the microbiology lab. Closed-toed shoes must be worn at all time. Thongs or sandals that you wear on your feet. A woman's fancy shoes that have an open-toed shoes are not allowed. If you do wear those to Clark College and you go into the microbiology lab, you must change those shoes either right before you enter the lab or while you're at the cubbyhole. And then put your sandals, for example, in the cubbyhole. Lab coats are required while you're in the microbiology lab. They must be worn completely buttoned whenever you're in the lab. We do have lab coats for you to borrow, and you would generally use that same lab coat all term lab, but you may also purchase a lab coat from the bookstore, and then in the first lab, bring it in. And usually you put your name on it, and then it'll be stored in the lab all term long until the end of the term, which you would then turn it in to the instructor. And you don't get it at that point, but the instructor would then set it aside, and then the lab prep people would take it and then autoclave it to sterilize any microbes, wash it and dry it, and then hang it up for you to pick up sometime after the term. Usually it's picked up between terms. Any question about any of that? Most students just use the lab coats that we provide in the lab, and they too would be autoclaved and washed in between terms. So you start with a clean, fresh lab coat with no bugs on it.
I had a quick question.
Uh-huh.
So the lab attire, on the worksheet, there was a question that asked what to wear before and then after you get into the lab. And I was kind of confused on the after part, or if it was part of the same answer of the first one.
Well, let me ask you this. Would you put something on before you enter the lab and then put the same thing on after you enter the lab?
No, but that's why I was kind of confused.
Okay.
Could we go over the picture kind of pointed also at like the face, and I didn't know if it was the goggles or hair being
I
pulled
will
back.
get to that. Okay.
Okay.
Okay. So when I get to it, you can ask your question at that point, but we're not there yet.
Oh,
All right. All right. So I will get to that. So lab coats are never removed from the lab. They're always kept in the lab room for the duration of the term. And then they would only be removed after the term is ended, actually. Goggles are required anytime students are working with a broth liquid culture, which has microbes in it, or whenever there's a splash hazard. So if you're working with a liquid culture that has microbes in it, you could splash it into your eye, so you have to wear goggles. And also if you're working with, generally the only thing we have in the microbiology lab is a stain. And if the stain is hazardous to your eyes, you have to wear goggles while you're doing the stain, for example. Like the Gram stain has, I think, 2 ingredients, which would be hazardous to your eyes. So you have to wear goggles when you're doing the Gram stain. At most times in the lab, you're not going to be working with a splash hazard, so you would not be working with goggles. Goggles are supplied by the lab. And so what you would do is put on your lab coat, usually put on your gloves, and then you go over to the cabinet where the goggles are, and then you pick out goggles which are sterilized by UV light. And then when you're done with the goggles, usually when you're done with the lab or you're done working with the splash hazard, you can put the goggles back. Let's see, where do you put them? You put them in the dirty goggle place, and then the instructor at the end of the lab will put them in the cabinet and then turn on the UV light in the cabinet to sterilize the goggles. Cleanliness is a virtue in microbiology, so always when you come into the lab, before you begin any work, you disinfect your lab bench before you begin working. And then after you leave, or after you're done, you disinfect the same area of your workbench before you leave. So you're always disinfecting before you begin the work and then after you've done your work. How you do that is we have bottles of disinfectant. You spray it down over your work area. You grab your paper towels and then smear around the disinfectant and then wipe it up with the paper towels, leaving a little bit of moisture on the lab bench to air dry. You should note where the fire extinguisher, the fire blanket, the eye wash station, the first aid materials are located. You don't need to know that, but you should know that we have safety equipment in the lab, including a fire extinguisher, a fire blanket. We have a fire alarm as well, an eye wash station, a shower, and then the first aid material where you got band-aids and other things like that. Accidents should be reported to your instructor. Minor accidents should be cleaned as follows. Always clean broken glass first. Broken glass can cut your skin, including if you have gloves on. And if there's microbes in it, you don't want that to happen. So always use a brush and a broom and a dustpan to pick up broken glass. Never use your fingers. Dispose of broken glass in the proper receptacle. We have a receptacle for putting broken glass. We don't use strong acids or bases, but they should be diluted with a neutralizer and then wiped up quickly. Spilled bacterial culture should be cleaned as follows. Cover the spilled material with lots of paper towels. Drench the paper towels with disinfectant. Let the disinfectant sit for 15 minutes before wiping up with paper towels. Be especially careful if you have broken glass in it. Put the broken glass in the broken glass container, everything else in the autoclave container. Wipe up the area with more dry paper towels. Discard the paper towels in the biohazard trash can. Any questions about cleaning accidents? Be sure to keep hair and paper away from the Bunsen burners or the back-to incinerators. Both of these should be turned off when they're not in use and before you leave the lab. Mouth pipetting is not allowed. Never take cultures out of the lab. Samples can only be brought into the lab upon authorization by the instructor. If you have a cut on your hand, you should go through this procedure. Wash your hands normally, go over to the first aid kit, apply a Band-Aid, and then put a glove over the Band-Aid. As always, when you're done, take off the gloves, go wash your hands. You can go grab another Band-Aid if you want, and then leave the lab. All microbial cultures must be completely labeled with your name, the date, the nature of the specimen, and the medium used. The temperature is optional. Old specimens must be discarded properly in the following ways. Agar plates should be discarded in the appropriately labeled trash can. So this would be the biohazard container. Glass slides and cotton swabs should be put in a glass container containing disinfectant, and they're found in the rear of the lab. We also have a cotton swab container on each table. You don't need to know that, but pipettes should be placed in the appropriately container. If plastic pipettes and they're not contaminated, they may be placed in the trash can. If they're contaminated and they're plastic pipettes, they should go in the biohazard container. If they're a glass pipette, they should go in the used, dirty glass container for recycling. Not recycling, but for cleaning. Glass test tubes should have the label tape removed and then should be placed upright in a test tube rack in the discard bin, which is in the rear of the lab. You don't need to know the rear of the lab. Supplies needed for the current lab will be found at the front of the lab. Each pair of students will be assigned a drawer under the lab benches. Your assigned drawer will contain lab supplies like paper, pencil, pens. Do not use supplies in other drawers. You may store things in your assigned drawer, but there is no ability to lock the drawers, so don't leave valuables in your drawers. So remember the area where your coat and your bags are stored, your book bags are stored, and the drawer is considered outside the lab. To leave, you must remove your lab coat and your gloves, wash your hands, and you follow the same procedure before you enter the lab. Meaning you come into the lab, you wash your hands, put on your lab coat, put on gloves. So to enter the lab, you got to walk into the door, past the cubbyhole, and then just walk a few feet into the lab. To leave the lab, you walk to the cubbyhole. Obviously, you do that after you wash your hands. Any questions about any of that? All right. If not, let's talk about Lab 00, lab safety training. I didn't shut that down. Let me shut that down.
Did you want me to come back to my question about before and after, or?
I haven't gotten to it yet. It's in the lab.
Okay. Oh, okay.
Ubiquity, oops, that's the wrong lab module. I guess that's it then. All right. Excuse me here. All right, you're not going to be working the lab, but you do need to know this material as many of these rules are relevant during your career. You also know these rules for your quiz. The learning objectives of Lab 00, lab safety, is 1, be able to distinguish between safe and unsafe laboratory practices, understand where relevant safety equipment is located in the lab, and 3, understand how to safely dispose of contaminated material. Microbiologists have a number of lab safety rules that must be followed while working with potentially pathogenic microorganisms. You're not going to be actually performing any of these activities, but it is important to understand the guidelines to create a safe laboratory environment. So you must wear proper attire, which in the lab, you must put on a clean lab coat that covers your lap. When sitting, it should be totally buttoned. You must wear disposable gloves. You must wear closed-toed shoes. If you're working with a splash hazard, you must wear goggles. Lab coats, goggles, and gloves must be removed before leaving the lab. And remember, you can pick up all these things in the lab. So these items are things you put on after you enter the lab. Now, what would be something you put on before you enter the lab for safety? Nobody's
You'd
gone.
already have closed-toed shoes on?
Yeah, you should have closed-toed shoes. You should have clothes on, pants and a shirt. If you're wearing goggles, that's not goggles, glasses. That's not exactly safety equipment. But if you do wear glasses, you can bring them into the lab. So those would be things that you should put on before you enter the lab. And it's not stated in this lab directly, but it's shown in this picture here. Long hair should be tied back. We have a hair tie or a hair net. And I think this student is actually wearing a hair net. Absolutely no food or drink brought into the lab or any electronics. Remember, if you have these things, they go in the cubbyhole, which remember is considered outside the lab. Any question about any of that? Now, obviously, you should not go with your gloves, open your backpack, grab a snack to eat while you're inside the room. What you should do if you wanted something to eat or drink, you should take off your gloves, take off your lab coat, go wash your hands, go grab your food from the backpack, and then you should leave the room to go and eat it. And then you come back in, put anything you haven't eaten in your backpack, and then go to the sink, wash your hands, go put on your lab coat, and then go put on your gloves. All personal items are stored in the personal storage area immediately past the main lab door, meaning right next to the door, and this region is considered outside the lab. Personal items that are brought into the main part of the lab will be confiscated and returned only if they've been soaked in bleach for 15 minutes or autoclaved. And that'll destroy any electronics. So that's what somebody should be looking like in the lab. And that's actually close to what our lab looks like. Well, that's not exactly a good picture because that's not our lab. Here's a picture of our lab. Keep your work area clean and uncluttered. Always disinfect the lab station. Like if you're going to work right here, you disinfect this whole area right there with disinfectant. And there's a disinfectant. And then move it around with paper towels, which you can't really see much. There's a Bacto incinerator right there. There's a microscope. No paper towels, no disinfectant, which they must have cleared that area from there because we usually have disinfectant on the lab bench. Of course, it could be right here. Discard waste in the appropriate receptacles, regular trash, non-sharp objects, meaning paper towels, gloves that are not contaminated, can be put in the trash can. Never put glass in the trash can. Never put sharps in the trash can. Biohazard container, contaminated paper towels, anything that's contaminated with bacteria, non-reusable agar plates, and that's in a plastic container, put in the biohazard container. You never put glass in the biohazard container. So no broken glass, no sharps, which we don't really use sharps in microbiology, but none of that in the biohazard container. We have contaminated test tube bins for putting glass test tubes, for disposing tubes, broth, and augers. We have a bin for glass pipettes. Put the glass pipettes, if they've been used, in there. And you always put glass pipettes there once you've used them, because even if you're not contaminated, we'll always wash them and reuse them. Soiled swabs, slides, cover slips go in different containers. For each, we have a usually a container on the lab bench as well as at the back of the lab for soiled swabs. For cover slides, we put them in a dish with slides that have disinfectant in it. The slides will be washed and then, well, I guess dried and then put for reuse. The cover slips we don't reuse. They will be put in disinfectant and then discarded. So all materials that contain contaminated or potentially pathogenic organism must be just sterilized prior to use or disposal. Ask your instructor if you're not sure what to do. And there's a biohazard container, usually has a plastic thing. Put the stuff in there. The plastic bag will be removed, autoclaved, before it will then be disposed in the regular trash. And that's to kill all the microbes. Once again, report all spills and accidents immediately, no matter how slight. Cover the material with paper towels and then drench the area with disinfectant, allowing it to soak for 15 minutes to kill the microbes. And then clean it up with clean, dry paper towels. If there's no glass, put it all in the biohazard container. If there's glass, you need to separate out the glass using a dust pan and a small broom, putting the glass in the broken glass container, and then all the paper towels, and then the wiped up area, put it in the biohazard dish. Biohazard trash. Keep lids on all reagents when they're not in use. Do not pour reagents back into a container. Don't ever pour chemicals down the drain. We usually have recycling for where to put chemicals. We don't put them down the drain. And then they'll be disposed of appropriately. No mouth pipetting ever. No biting your nails. No biting pencils while you're in the lab. When a Bunsen burner or Bacto incinerator are not in use, turn them off. Realize the Bacto incinerator will take 5 minutes to turn on to become red hot. So you might want to turn it on 5 minutes before you want to use it. And then when to use it, what you do is you put the loop or whatever you're using in the flame or the Bacto incinerator until it becomes red hot. You can kind of see it right there. Let's just sterilize the loop. And then you cool it and then pick up the cells or organisms you want. If you're familiar with the safety equipment we have in the lab, we have a fire alarm, we have a fire blanket. I'm not sure if that's a fire blanket or the hose. Let's assume that's the fire blanket. A shower, which if you ever catch on fire, you can turn on the shower. Don't do that if you ever go into the lab because it just puts the water on the floor. It's true we have a drain in the floor, but it'll make a mess out of the area underneath the shower there. And then an eye wash station. A first aid kit. I'm not sure the fire alarm there. Oh, and there's what do you call that? A fire extinguisher. So all of this equipment is in the lab. Fortunately, the only time I've ever used any of them is the eye wash station. And this actually happened when I was a graduate student. A doctor splashed some chemical into his eye and he asked me what to do. And I said, go to the eye wash station. I took him to the eye wash station and then turned it on and then put his eye under the eye wash station. It's the only time I've ever used it. Never used it as an instructor, but it's there in case we need it. And obviously you guys will be all online, so you just need to know what safety equipment is in the lab. You don't need to know where it is in the lab because you'll never go into the lab. All right, so complete the laboratory exercises in a separate file. This goes over the lab safety procedures and safety rules, which I've already discussed because it's in my syllabus. All right, let's go to the questions. So for turning in the worksheet, you need to get the worksheet. And for today's worksheet, it's lab worksheet 0 to lab module 00. It'll probably just say worksheet 0 or 00. Fill in an answer to each asterisk. Don't remove the asterisks. It'll only confuse you if you do so. I remember how many questions or how many answers I'm expecting in each rule or question. So rule 1 in the lab, what do you always wear for safety before you enter the lab? This is not directly stated in the lab, but it is shown in a picture. And the picture is
Did you want us to say it or just wait?
no, you don't need to say an answer. If you have 4 items, just fill it in. It's shown in this picture. Now realize you can't see much of her shirt. So hopefully that's something you put on before you enter the lab. Remember, you have to have 4 answers. For rule 2, after you enter the lab, what 2 items do you put on to keep yourself safe in the lab? And obviously, this is something you do after you enter the lab, not before you enter the lab. You only need to answer 2 things there. Rule 3, look up what a safety data sheet is online. Each chemical in the lab has a safety data sheet, SDS, that describes things about it. Provide at least a phrase for each answer, and then tell me 1, 2, 3, 4, 5 different items you can find in the lab safety data sheet online. Rule 4, name 5 items of safety equipment found in the lab. Rule 5, name 2 items you can take into the lab. Remember, anything you're wearing, you can take into the lab. Can you eat or drink in the lab? Put that in asterisk 3. 2, list 1 item of laboratory attire that you must wear in the microbiology lab. 3, list 1 item belonging to you that is allowed to be brought into a microbiology lab. 4, list 1 item belonging to you that is not allowed in a microbiology lab. And then 5, submit this file by 8 a.m. Friday of week 1 in the folder to lab worksheet 00, or you'll be dropped from the class. I will use this for attendance for week 1. Any questions about lab 00? All right, go ahead and get started on lab 00. I'll be here to answer any questions until about 8 o'clock or until the last student logs out.
I do have a quick question.
Uh-huh.
The SDS, where are we supposed to be looking those up to know
Online.
what? Yes, which,
www.google.com.
how do we know what chemicals are in our lab?
You don't need to know that. You just need to know a general item that's in the safety data sheet. Like I'll give you a hint and say one thing is chemical disposal, meaning disposal of the chemical. that's told in the safety data sheet.
Oh, okay. So just give a phrase for each of the answers, meaning something that's found in there, and explain what it is.
Uh-huh.
Okay, thank you.
All right.
I have a question now. Now that can I share my screen to ask the question I was gonna ask in the lecture?
Yes, just a minute. I've got to change the share. I think I'm not sharing my screen at the moment. Let me change the properties. Oh, come on. All right, I changed it. Now I got to share the screen again. All right, you should be able to share your screen now. Oops,
I don't think so.
didn't work. It looks like it's showing something like I didn't click that, which isn't way, because it should be sharing all participant. Let's try that again.
I think
Trouble getting it to take, because it's showing it here. Let me close it. Try it now. See if that took.
no, it still says disabled share.
Well, I've got it here for all participants, but I don't know how to.
Wait, maybe I
Oh,
can't.
wait a minute. Let's go here, all participants.
Can you see my screen?
Just a minute. Is this your screen here?
Yeah, it says I'm sharing.
Uh-huh.
Okay, so when I go to the Respondus quiz and I press take quiz, and then I press download Respondus, and I already
You're
have
supposed
it.
to download Respondus Lockdown Browser before you take the quiz.
So how would I show that I did the quiz right here?
Like I said, you gotta download Respondus Lockdown first, and
I
it
do
looks
have
like you're
it.
trying to get to Respondus. Take the quiz inside a normal browser. Respondus Lockdown Browser is an independent browser. You have
I
to take
have
the quiz.
Respondus.
Okay, open it up and then go to the quiz with Respondus Lockdown Browser.
That's what it shows when I open it.
I'm not seeing, can you show me about, I'm not seeing, oops. I'm not seeing what you're showing me in. You got to
So like
show me about what this is. Meaning I'm only seeing the screen here. I need to see what you're in.
I'm showing you.
Hey,
So like
Francesca,
No,
might
click
be able
on
to help you out here.
it should say about, meaning someplace on the left or top. It should be about. I need to see that.
I'm so okay.
Hey, Francesca.
Yeah.
When I had to do something like this for my proctored essay, you have to close out of everything completely, click on the respondents lockdown browser, and then log into the Canvas, and then you should be able to take it.
But how do you log onto like how do you go into the lockdown browser and then go into a quiz? Because aren't you
I
locked
just
down?
told you, you have to be in Lockdown Browser to go into the quiz. So you have to go into the class with the Lockdown Browser. It's another browser.
I know that I hear that you're saying that, but what's frustrating is that we're trying to look for the other browser and we're not able to find it. And so
Well, you
how
got to
can
download the browser
we
first.
already downloaded? You're acting like we haven't downloaded. We already downloaded the browser. We
So
have
then
it.
open up the browser.
It's its own icon.
You're in another browser, is what I'm guessing. I can't see what you're in because you haven't shown that. But I'm guessing you're in another browser. You cannot take the quiz in another browser. It has to be Lockdown Browser only.
Probably the wrong browser.
So just shut down all your browsers and then open lockdown browser. And yes, you'll have to log into the class.
I have another question. When
Uh-huh.
should we expect these videos to be uploaded on YouTube?
The video will not be uploaded on YouTube until after the end of the day. And that's assuming I upload them right away. And then they'll take at least a half an hour, maybe longer, to put text elements in the video. And then after that, you should be able to view the video. If they're not up by a day after the session, you should send me an email as a reminder to post it. Sometimes I forget to post it, but usually I upload it after the lab. Meaning I haven't posted anything yet today. Well, let's stop recording.